students. So as I told you yesterday, today I will be discussing about the concept of linkage and how linkage is opposite of that of independent assortment. So if you recall Mendel's dihybrid cross, at the end of dihybrid cross, Mendel could come up with the idea of independent assortment, where the factors gets for different contrasting character, they get assorted independently. Now what do you mean by linkage? To understand this, we have to go to the chromosomal theory of inheritance. Chromosomal theory of inheritance means the segregation of genes or factors is actually dependent on the segregation or separation of the chromosome. Because, for example, suppose this is a pair of homologous chromosome. Homologous chromosomes are chromosomes which stays in pair in a diploid condition. Suppose there is a pair of homologous chromosome, and what are factors? Nowadays we know that factors are nothing but genes. So they will be located where? They will be located on the chromosome. Suppose we have two factors, let's say capital A and small a. Now this is the condition in the generation, let's say the parental generation. Now from here, when the factors will separate to form gametes, it is actually the chromosomes which will be separating. Now this entire thing is inside one cell. So from here, there is a diploid condition since the chromosomes are in pairs. From here, when gametes will be formed, separation of the factors is actually separation of the chromosomes. So one chromosome will come to each gamete. So along with the chromosomes, the factors are moving out. So actually movement of the factors or separation of the factors is actually separation of the chromosome. So this is the chromosomal theory of inheritance. In your NCERT book, you will find there is a description of chromosomal theory of inheritance. So you read that first before trying to understand this video. Now, what do you mean by independent assortment? Let's recall what we got in Mendel's dihybrid cross. In the, let's say there are factors for two contrasting characters, two pairs of contrasting characters, suppose for tallness and for seed shape, tall and dwarf, round and wrinkled seeds. Now, when this was the combination in the F1 generation, from here when gametes were formed, if you recall, Mendel predicted that, or he said that there will be 4 varieties of gametes which will be formed in this pattern. So from this he gave his law of independent assortment. Now next question is, if suppose this is the combination, from here can this be a probability that only capital T and capital R will come in one gamete and small t and small r will come in one gamete? Is this a possibility? The question is that. Now this was shown by Mendel. Now this is also a possibility provided we know where the factors are located and how they segregate during gamete formation. Now let us take two cases to understand the difference between when this will be a possibility and when this will be a possibility. Now suppose this is a pair of homologous chromosome having factor capital T and small t, and suppose there is another pair of homologous chromosome having capital R and small r, and they are inside a cell. So the factors are located in different pairs of homologous chromosomes. Now from this cell there is also a diploid cell because the chromosomes are in pairs. From here when gametes will be formed, suppose this is the first gamete, I am talking about the probabilities. Now from this pair, one chromosome will come. Let's say capital T, the chromosome carrying capital T has come here. And similarly, from this pair also one chromosome will come. Let's say the chromosome carrying capital R has come. So the combination in this gamete, the probability will be capital R, capital T, capital R. Similarly, it can also be the case that the chromosome for capital T comes in and the chromosome carrying small r comes in. So then the combination will be capital T, small r. Similarly, there may be a probability that the chromosome carrying small t comes in, and from this pair the chromosome carrying capital R comes in. So we'll get the combination small t, capital R. Similarly, there might be a possibility that from here small t comes in from this pair, and from this pair small r comes in. So the gamete will have a probability of small t and small r. So this is the concept of independent assortment, where the factors, when they are located for a particular contrasting character, when they are located in separate pairs of chromosomes, then they will segregate independent of the factors for other pair of contrasting character. And this is what Mendel discovered, and he was lucky indeed not to get confused, because all the 7 pairs of characters which he considered, the factors for all the 7 pairs of contrasting characters were located in different pairs of homologous chromosome. Okay, now let us understand when this will be the condition. Now suppose there is the cell, there is a pair of homologous chromosome, there is also a diploid condition since the chromosomes are in pairs. Now let's say the factors for tallness is located here. One chromosome may have hundreds and thousands of genes in it. So it's not that it will have only one gene as shown here. It may have more genes. Let's say the factors for seed shape is located in the same pair of chromosome in this way. Now from here, when gametes will be formed, what happens in gametes? Chromosome number becomes half. So there is just one pair. So let's say this chromosome comes over to this gamete. This is the probability. So what will happen? Capital T will be coming with capital R. So what we get? We get that this gamete will have a combination of capital T and capital R. And this chromosome coming over to this gamete, small t and small r, so this will be the probability. So this is the concept of linkage. Linkage means when the factors or genes controlling different characters are linked together. Linked together means they will be located in the same chromosome, so that when there will be gamete formation, when the chromosome will travel over to the gamete, all the genes located in that chromosome will travel together. So this is the concept of linkage. This is the concept of independent assortment. So the concept of linkage was discovered much later. Initial idea of linkage was given by W. Bateson, E.R. Saunders, and R. Punnett. And later on it was elaborated, it was established by experiments done by T.H. Morgan. Now Morgan's activity is there in your NCERT book, what he did with fruit fly. This linkage he could establish by working on fruit flies. Fruit flies, Drosophila melanogaster. You find small fly-like creatures sitting on cut fruits and vegetables. So that is called fruit fly. So experimenting with fruit flies, he could establish the concept of linkage. And in linkage also there is a table given in your book to show to what extent linkage occurs and what is the relationship between linkage and recombination. recombination. Now what basically happens is that, suppose this is a chromosome, this is one gene, let's say A, this is another gene or factor, let's say B, and let's say their alleles are here, let's say their recessive alleles are here. Similarly, if we consider another pair of chromosome, let's say A is here and B is here. So what is the difference we find? That the distance between these two genes are much lesser compared to these two genes. Here the distance is more. So what basically happens is that we have heard about, or we have studied about, a process which is called crossing over, which occurs during the Pectian stage. The Pectian stage of meiosis 1. So what happens in Pectian? Basically, there is a crossing over which happens, and the chromosomes, their segments are interchanged. Now when they will be interchanging, there will be shuffling of the genes. Now basically what happens here is that we find, if the distance between the genes are less, the amount of crossing over which will occur will be much lesser, and less crossing over means lesser recombination. Recombination is a result of crossing over, and the far away the genes are located, there will be more chances of crossing over, and more new combinations or more recombinants will come in. So if you follow in your NCERT book, they have given the example of crossing over of two genes in case of Drosophila. You don't have to understand what the genes are responsible for. You just go through the table, and at the end you will find where the genes are located close to each other, the amount of recombinants are less, and the amount of parental characters which are maintained through the generations is more. And on the other side, you will find that where the genes are located far away, at the end the number of recombinants which are arising is more, because there will be more crossing over. So this is an overall idea of linkage, how it is different from independent assortment, and how linkage determines the amount of recombination. So I hope if you have any doubts, you can clarify later. Thank you.